today's discussion. Today we shall be looking at cleavage, blastocyst formation, and twinning. At least by the end of this discussion, we should be able to know what cleavage is and the basic process that occurs during cleavage and how this process may result into twin pregnancy. We should be able to appreciate formation of Hydatidiform, which is an abnormality that is associated with a blastocyst.
Okay, let's now look at the sequence of events that occur during cleavage and formation of blastocyst. So cleavage itself is a series of mitotic division that occur immediately after fertilization. But two important things that I want you to note before we actually look at the series of events that occur. One is that during cleavage, the cells that are being formed are termed blastomere. Blastomere. So one thing that you need, or two things that you need to note about this blastomere, they increase in number with each successive division. However, the overall size of the zygote remains constant. Increase cells, but size of zygote remains the same. Another thing that is important to note during this cleavage, as the cell numbers are increasing, the size, the overall size of the cells also reduce. So for example, you can see this is one cell state, this is two cell state. The cells are smaller than here. This is four cell state. The cells are smaller than here. So after fertilization, we form what we call a zygote. This zygote has 46 chromosomes. The zygotes divide, or it has entered into a series of mitotic division and form two cells. Or this is two cell stage. These two cells also divide and forms four cell stage or four cells. The zygote, you will see that these four cells are smaller than the two cells. Then the four cells also divide to form 8 cells. Now the 8 cells divide to form the 16 cells. Now this 16 cell stage is termed morula. Morula and morula. So this morula is in the uterus. In other words, this process occurs as this zygote that has been formed is moving through the fallopian tube. Now the cells of the morula, or in the morula stage, are smaller and their cytoplasm is too small and it hardly supports the cellular activity. Now what occurs, or what happens to this morula? This morula is in the uterus, and in the uterus there is what we call the uterine fluid, what we term uterine milk. Now what happens is the uterine milk begins to diffuse inside the morula. The fluids, the uterus, begin to diffuse inside. So as more and more fluids diffuse and begin to occupy the intracellular space, the outer cells is pushed to the membrane. And then the inner cells, or the cells which were initially in the center, is also pushed to one pole. Now the pole that has been formed is termed the embryonic pole, and these cells which have aggregated at the embryonic pole are called the embryo, blast, or inner cell mass. And then the ones which has been pushed to the periphery, which has become more and more flatter, are termed as the outer cell mass. Now, this outer cell mass is also termed as trophoblast. Then there is a space which is filled with fluid that has been formed here. This space that is filled with fluid is what we call the blastocele. We call this one blastocele. And now, what we have formed, the whole of this structure, is what we term as the blastocyst. Okay, we already looked at how cleavage and blastocyst formation occurs. If I just do an overview on structure of the blastocyst, this is our outer cell mass, which is also known as the trophoblast. This is the component that results into the fetal component of the placenta. Then this one is the inner cell mass that we term embryo blast. This component will develop into the actual embryo, and of course this space is the blastocyst, and the overall structure is termed as a blastocyst. Now, there is a very common abnormality that is associated with blastocyst, and this abnormality is termed as a blighted blastocyst. A blighted blastocyst. So a blighted blastocyst is as a result of abnormality in fertilization. So normally, during fertilization, one sperm fertilizes an ovum to form a zygote. However, in a blighted blastocyst, we shall have either two sperms fertilizing an ovum, or one sperm fertilizing an empty ovum. So what happens? This is a blighted cyst. Now, when we have a blighted cyst, which results either due to fertilization of an ovum with two sperms or an empty ovum with one sperm, then we form a blighted blastocyst. That is after undergoing a series of cleavage. Now, this blighted ovum does not proceed to grow into a fully formed fetus. What happens? The embryoblast degenerates. This embryoblast degenerates. After degeneration of the embryoblast, the trophoblast begins to undergo proliferation, and it forms an avascular structure, which is grape-like, and that is what we term as molar pregnancy, or a hydatid mole. You can see hydatid mole. Okay, finally we shall look at what we call twinning. Twinning is development of two embryos within the uterine cavity. We have two types of twinning. One is dizygotic twinning, and the other one is monozygotic twinning. During fertilization, a sperm plus an ovum forms zygote. Now, when we have two ovum release during ovulation, what we normally term as hyperovulation. We have two ovum, two ovum. So this ovum will be individually fertilized by different sperms. This one will lead to formation of what we call dizygotic twins. In other words, these twins are non-identical. These twins may be of the same or different sex. They may also have different physical attributes. Then the other kind of twinning is the monozygotic twins, which is actually related with cleavage. Monozygotic twins or monozygotic twins. Now, we already looked at what we call cleavage and the process or the sequence of events that occur during cleavage. We discovered after fertilization, we form what we call a zygote. Now, this zygote enters into series of mitotic division, as we already saw. One cell formed 2, 2 formed 4, 4 formed 8, 8 formed 16, 16 is the morula, which later on formed the blastocyst. Now, during this series of events, what we call cleavage, there might be a complete splitting of the embryo, and when this splitting occurs, each of the components of the embryo results into a different fetus, and that is what we call monozygotic twins. So we have basically 4 types of monozygotic twins. One, we have what we call dichorionic, diamniotic twins. The second one, we have what we call monochorionic, diamniotic twins. The third, we have what we call monochorionic, monoamniotic twins. And then finally, we have what we call the conjoined twins. As I already mentioned, that these twins result into splitting of the embryo into 2. In other words, these twins are actually clones. So how do you differentiate, or how do I tell which is which? Now, when splitting of this embryo occurs much earlier during the process of cleavage, the splitting can occur within the first 2 to 3 days after fertilization. Within the first 2 to 3 days during fertilization, the embryo will completely separate, and this embryo will form different placentas and different amniotic sac. And that kind of twin, we term them as dichorionic, diamniotic twin. Then the splitting may also occur slightly later. When the splitting occurs from 3rd to 8th day, that one is around, when the splitting occurs within the 3rd to the 8th day after fertilization, we shall have the embryo formed, or the fetus that are being formed. They will have the same placenta and different amniotic cavity. So that kind of twins are called mono-di twins. Then we may also have the splitting or separation of this twin occurring in the 2nd week, that is day 8 to day 13. During the 2nd week, we have blastocyst already formed, and then we are having what we call the bilaminar disc already formed. However, this splitting occurs before the formation of what we call primitive streak. When this separation of the embryo occurs here, we shall form what we call monochorionic, monoamniotic twin. In other words, the same placenta, the same amniotic cavity. However, the splitting may slightly delay, maybe occurring day 13 and above. There, it will lead to incomplete separation of the fetus that are being formed. If there is incomplete separation of this fetus, we shall have what we call the conjoined twins. Thank you for your time. See you in the next episode.